Now listen. Sir, when we try to insert or when we try to introduce these vectors into the host bacterial cell, now we are trying to introduce this now we are trying to introduce these vectors into the host bacterial cells. Three types of cells are obtained. How many types of cells are obtained, sir? Three types of cells are obtained. When we try to introduce these vectors into the into the host bacterial cells, three types of cells are obtained. How many type of cells are obtained? Three types of cells are obtained. When we try to introduce these vectors. Sir, what is the meaning of these vectors here? Altered vector, unaltered vector. Altered vector, unaltered vector. Into what? Host bacterial cells. Then three types of cells obtained. How many type of cells are obtained? Three types of cells are obtained. Now, sir, what are the three type of cells? Now we are going to see the three type of cells. Now, listen this statement. Sir, here how many type of vectors we are having? First you tell me that. How many type of vectors are there? Two types. What are they? Altered vector. Second one is unaltered vector. Now we are trying to introduce these vectors. We are trying to introduce these vectors into what, sir? We are trying to introduce these vectors into the host bacterial cell. Yes or no? Then what happens? Listen. Sir, this is which vector, sir? For example, this is altered vector, right? For example, this altered vector has entered into one bacterial cell, sir. Altered vector has entered into one bacterial cell, sir. Which vector entered, sir? Altered vector. Altered vector has entered into one bacterial cell. Now you tell me, sir, altered vector means it is having ampicillin antibiotic resistant gene, but it is not having tetracycline antibiotic resistant gene. Having ampicillin antibiotic resistant gene, not having tetracycline antibiotic resistant gene. Yes or no? Right? Now, when we are trying to introduce these vectors into the host bacterial cell, this vector has entered into another host bacterial cell. This vector has entered into another, what's our host bacterial cell. Into this host bacterial cell, what has entered, sir? Unaltered vector. Into this unaltered vector has entered. Sir, unaltered vector means what, sir? It is having ampicillin antibiotic resistant gene as well as tetracycline antibiotic resistant gene. Is it okay? Right? Now go for the third case. Sir, this is another what? Host bacterial cell, sir. Into this host bacterial cell, neither altered vector nor unaltered vector has entered, sir. Therefore, no vector entry. No vector entry. It means no vector has entered into this. Is it okay? Sir, we are trying to introduce these vectors. We are trying to introduce these vectors into the host bacterial cells, sir. Then how many type of cells we got? Three type of cells we got. It means only three possibilities. Number one, altered vector may enter into a host bacterial cell. Second possibility, unaltered vector may enter into the host bacterial cell. Third possibility, neither altered vector nor unaltered vector enters. Is there any fourth possibility? No. Therefore, when we try to introduce the right vectors into the host bacterial cells, only three possibilities. Either altered vector enters into the host bacterial cell, or unaltered vector enters into the host bacterial cell, or vector does not enters into a host bacterial cell. How many type of cells we got? Three type of cells. The same statement I wrote here. Is it okay? Now you tell me, sir, this host bacterial cell has taken up vector, taken up DNA, yes or no? It means undergone transformation. Undergone what? Transformation. Therefore, this is which cell, sir? This is a transformed cell. This is which cell, sir? Transformed cell. Sir, this host bacterial cell has taken up vector, has taken up the DNA. Therefore, this is also transformed cell, sir. But this has not taken up a vector. This has not taken up the DNA. Therefore, what is this one, sir? This is a non-transformed cell. This is non-transformed cell. This is which cell, sir? Transformed cell. But this transformed cell is having which vector? Altered vector. What is another name for altered vector? Recombinant vector. So we are calling this transformed cell as a recombinant transformed cell. Sir, this is also transformed cell, sir. But this transformed cell, is it recombinant? No. Therefore, this cell becomes a non-recombinant transformed cell. What it becomes? Non-recombinant transformed cell. Therefore, how many type of cells we got? We got three types of cells. What are the three type of cells, sir? Number one, recombinant transformed cells. Okay. Number two, what is the second one, sir? Non-recombinant. Non-recombinant but transformed, right? Non-recombinant transformed cells. Number three, non-transformed cells. Therefore, we got three type of cells. Number one, recombinant transformed cell. Recombinant transformed cell is with which vectors are? Altered vector. Recombinant vector. Okay, right? Non-recombinant transformed cell, right? With unaltered vector. With what, sir? Unaltered vector. With unaltered vector or non-recombinant vector. Whereas a non-transformed cell without vector. No another possibility. Only these three possibilities are there. Is it okay? Right? Sir, altered vector. Altered vector means it is with or without ampicillin. It is having ampicillin what? Ampicillin antibiotic resistant gene. But not having what's our tetracycline antibiotic resistant gene. Whereas unaltered vector, right? Unaltered vector, it is having ampicillin antibiotic resistant gene as well as tetracycline antibiotic resistant gene. Both are present, right? Right. Whereas without a vector we are saying, it means here ampicillin antibiotic resistant gene is absent as well as a tetracycline antibiotic resistant gene is also absent. Therefore, recombinant transformed cell contains altered vector. So, recombinant transformed cell contains ampicillin antibiotic resistant gene, but a tetracycline antibiotic resistant gene is absent. Non-recombinant transformed cell contains unaltered vector. So, non-recombinant transformed cells contains ampicillin as well as tetracycline antibiotic resistant genes. Non-transformed cell neither contains recombinant vector nor contains non-recombinant vector. So, it is without ampicillin and without tetracycline antibiotic resistant genes. Therefore, when we try to introduce these vectors into the host bacterial cells, how many possibilities? Three possibilities. altered vector may enter or unaltered vector may enter into the host bacterial cell, or vector may not enter into a host bacterial cell. These are the three possibilities. So we are getting three type of cells: recombinant transformed cells, non-recombinant transformed cells, next one is non-transformed cells. It's very lengthy topic, but it's very easy. If you concentrate, you will get easily. Is it okay? Clear? Right. Now listen here, sir. First we got three type of cells. No, first we will separate transformed cells from non-transformed cells, sir. First we are separating transformed cells from non-transformed cells. Okay, right. How we will separate? Right. First selection of transformed cells from non-transformed cells by growing all three types of cells on ampicillin antibiotic containing medium. Sir, first what we are doing? We are selecting transformed cells. From which cells, sir? From non-transformed cells. How? By growing these three type of cells. Where? On ampicillin antibiotic containing medium. How, sir? Listen first. Sir, first three type of cells, right? What is the first one, sir? First one is a recombinant transformed cells. What is the second one? Non-recombinant transformed cells, right? What is the third one? Non-transformed cells. Which cells, sir? Non-transformed cells. Now listen, what we are doing, right? We are growing these three cells, right? On what, sir? We are growing these three type of cells on ampicillin antibiotic containing medium. Grown on ampicillin antibiotic containing medium. Now you tell me, sir, recombinant transformed cell with ampicillin, without ampicillin. Recombinant transformed cell is having ampicillin antibiotic resistant gene. Therefore, recombinant transformed cell will survive. Sir, non-recombinant transformed cell. Non-recombinant transformed cell is also having ampicillin antibiotic resistant gene. Therefore, non-recombinant transformed cell will also survive. Now go for the third one, non-transformed cell. So do this non-transformed cell having ampicillin antibiotic resistant gene? No. Therefore, when grown on ampicillin antibiotic resistant gene, will it survive? No, it won't survive. Therefore, what happens to this non-transformed cell? Non-transformed cell will die. Therefore, what are selected? Transformed cells are selected from non-transformed cells. Selection of transformed cells. Which transformed cells? Recombinant transformed cells, non-recombinant transformed cells. Therefore, transformed cells are selected from non-transformed cells by growing them on what? Ampicillin antibiotic containing medium. Is this statement okay? Right? By using ampicillin antibiotic resistant gene, right, we selected transformed cells from non-transformed cells. Now, which cells are available, sir? Now, transformed cells are available. But transformed cells are of how many types? Two types. What are they? Recombinant transformed cells, non-recombinant transformed cells. Which cells we want? Recombinant transformed cells. Why? Recombinant transformed cells means contains recombinant vector. Recombinant vector means it contains desired gene. We want that desired gene. Yes or no? So, right, we have to select recombinant transformed cells from non-recombinant transformed cells. Is it okay? Right? Therefore, recombinant transformed cells. Recombinant transformed cells are selected. Recombinant transformed cells are selected from what, sir? Non-recombinant transformed cells by growing them on what, sir? Tetracycline antibiotic containing medium. Is it okay? Right? Therefore, recombinant transformed cells, right, they are selected from non-recombinant transformed cells by growing right, cells on what's a tetracycline antibiotic containing medium. See this statement carefully. Transformed cells are selected from non-transformed cells by growing on ampicillin antibiotic containing medium. Recombinant transformed cells are selected from non-recombinant transformed cells by growing them on tetracycline antibiotic resistant medium. Is this statement clear? Okay, right? Three type of cells, right? Type of cells. Grown on ampicillin, grown on tetracycline. What are the three type of cells? Number one, first one is recombinant transformed cells. Second one is non-recombinant transformed cells. And third one is non-transformed cells. These are three type of cells, right? Recombinant transformed cell having ampicillin antibiotic resistant gene, therefore survive when grown on ampicillin, but cannot survive when grown on tetracycline because it is recombinant, loses tetracycline, right? Non-recombinant transformed cell having ampicillin as well as tetracycline, so it is going to what, sir? Survive. Sir, non-transformed cell neither having ampicillin nor tetracycline, therefore dies. Is these statements okay? Clear? Therefore, three type of cells we got. What are they, sir? Recombinant transformed cells, non-recombinant transformed cells. Now listen the statement carefully. Sir, we are discussing by using two antibiotic resistant genes. Yes or no? Now listen. Selection of recombinants from non-recombinants. Selection of recombinants from non-recombinants by inhibition of antibiotics. Right, requires simultaneous plating. Requires what, sir? Simultaneous plating on two plates with different antibiotics. Once plating is done, right, on ampicillin, and next plating is done on what's our tetracycline. Therefore, this is a cumbersome process. Cumbersome process means hard to carry out. Why, sir? Because your plating should be done how many times? 2 times on 2 different plates having 2 different antibiotics. Yes or no? Right? Selection of recombinants from non-recombinants. Selection of recombinants from non-recombinants, right? Selection of recombinants from non-recombinants by inactivation of what? Inactivation of antibiotics. Inactivation of antibiotics is a cumbersome process. Is which process, sir? It is a cumbersome process. Why, sir? Why? As it requires simultaneous plating, simultaneous plating on 2 plates with different antibiotics. Is this statement okay? See, first we are growing on ampicillin, then we are growing on tetracycline. Therefore, plating, right, on 2 plates with different antibiotics. So we are calling this process as a cumbersome process. Cumbersome means hard to carry out. So alternate procedures or alternate techniques for selection of recombinants from non-recombinants were developed. Is it okay? Therefore, so what's our alternative techniques were developed. So alternative techniques were developed for selecting what? Recombinant cells, which are based on what are based on these alternative techniques which were developed for selecting recombinants, right? Which were based on blue-white selection. Which were based on what, sir? Blue-white selection, which were based on blue-white selections. Is it okay? Clear, right? Therefore, we completed second method. What it is? By using 2 antibiotic resistant genes. Right? Thank you.